Hey guys! This is a video presentation by your classmates Carlos, Alice, and myself, Zukra. Today, we are excited to talk about the nasty Clostridium difficile epidemic. Now, before we go into our catalyst paper, let's talk about what exactly is C. diff. C. diff is a bacteria that infects the colon of patients and can cause symptoms such as diarrhea, fever, and belly pain. Essentially, this thing could become fatal. And this is where our catalyst paper comes in. We had the pleasure of reading Dietary Trehalose Enhances Virulence of Epidemic Clostridium difficile, in which the authors zoom in to two ribotypes, RT027 and RT078. Both are epidemic C. diff ribotypes that apparently exhibit a competitive advantage over other strains in vitro and in mouse models of the C. diff infection. So why do the two ribotypes, RT027 and RT078, are better at reproducing and growing? Why do they have increased fitness? The amazing discovery is that both strains, unlike other ones, can use low concentrations of the disaccharide molecule trehalose as a sole source of carbon. Therefore, the scientists of the article propose that the implementation of trehalose as a food additive into the human diet shortly before the emergence of the two epidemic lineages helped select for their emergence and contributed to hypervirulence.
In order to understand how C. diff evolved to efficiently metabolize trehalose, we first need to investigate the process of how C. diff breaks down energy. In this case, it's via glycolysis. We will go into detail later on how RT027 and RT078 uniquely breaks down trehalose, but the key here is that both strains will undergo glycolysis. And I'm going to briefly explain the process of glycolysis. And first, we start with a glucose molecule, or 1 unit of glucose, and ATP will then come in, phosphorylate it, and then you will have glucose 6-phosphate. Trehalose utilizes this inducible operon pathway to create an enzyme to metabolize glucose. At default state, the active TREA repressor prevents the expression of the gene TREA, which encodes for an enzyme that is necessary for the metabolism of trehalose. So therefore, no RNA is made. So we essentially need a trehalose molecule to bind to the receptor and inactivate it, which is here. Trehalose will come in and bind here. And therefore, transcription can happen. So now Zook will go ahead and elaborate more on the RT027 strain.
What they found about the RT027 strains is that they have an overwhelmingly higher TREA gene expression than their counterparts. And why is that? They basically found SNPs in the TREA gene of all RT027 strains. The SNP encoded for a single amino acid substitution near the predicted binding pocket of TREA. The amino acid substitution went from being leucine to isoleucine. And this specific mutation increased the affinity for trehalose. Remember that an induced operon would need to suppress the repressor in order to allow transcription to go on. Because the affinity of the TREA repressor for trehalose increased, it allows the RT027 strains to be able to metabolize trehalose in very low concentrations. Thus is the reason for why they can proceed to grow under low levels of this disaccharide molecule.
Unlike the RT027 strain, the RT078 strain does not have a mutation in its repressor gene region of the TREA operon. Instead, this guy has a 4-gene insertion in its genome that gave it a new trehalose-specific phosphotransferase system, including the very important trehalose transporter, PTST. We know from biology that PTS is a distinct method used by bacteria for sugar uptake. It is a multi-component system that involves enzymes of the plasma membrane and the cytoplasm. Here we have a bacterial cell. Here is my molecule of trehalose outside of the cell trying to make its way through. Luckily, we have a new PTST transporter protein in the membrane that facilitates trehalose to enter the cell. From here on, this molecule of trehalose is available to bind the TREA repressor, enabling transcription to go on. Bacteria prefer glucose over any other nutrient. Glucose always comes first, but this trehalose-specific PTS system, and this transported protein is now available to help Trahalo's make its way into the bacterial cell. And this is how the RT078 is able to metabolize Trahalo's on low levels.
RT027 and RT078 have not always been epidemic. They've evolved to metabolize low levels of Trahalo's, and that is via convergent evolution. So, what the hell is convergent evolution? Well, convergent evolution is the independent evolution of similar features in a species.
So the big picture is this. Because we humans have recently been adding a lot of tree halos in food, it led to the emergence of two epidemic ribotypes that have evolved to become very sensitive to this disaccharide. The article states that this evolutionary pathway impacts the disease severity by causing an overproduction of toxin B, which can potentially be very fatal.
Thank you for listening.
Thank
Thank
you.
you.
Woo!